Hey guys, welcome to another episode of the Human AI podcast. Today we're going to talk about glycolysis, electron transport chain, glucose homeostasis, and glycogen metabolism. Few topics. I'm here with Grok, who's going to be helping us out, trying to explain these concepts. Take everything that Grok says with a pinch of salt. I hope you find this information enjoyable and also you find it informative. Okay.
Thanks for the sentiment. I'm ready to dive into whatever you're curious about. Any specific topic or question you want me to tackle?
Okay, so let's start with glycolysis. Grok, give us a good introduction on glycolysis. Explain us what it is. Why is it important?
All right, let's jump into glycolysis. It's a fundamental process in cells where one molecule of glucose, a 6-carbon sugar, gets broken down into 2 molecules of pyruvate, each with 3 carbons. This happens in the cytoplasm, doesn't need oxygen, and kicks off energy production in both aerobic and anaerobic conditions. Glycolysis is crucial because it's the first step in extracting energy from glucose, generating a bit of ATP. Think of it as the cell's energy currency. And NADH, a molecule that helps shuttle electrons for more energy production later. It's like the cell's starting engine, powering everything from muscle movement to brain function, and it's universal across nearly all living organisms. Want me to break down the steps or focus on something specific about it?
So what does metabolism have to do with glycolysis?
Metabolism is the big picture, all the chemical reactions in a cell that keep it alive, growing, and functioning. Glycolysis is a key part of metabolism, specifically in the subset called catabolis, where molecules like glucose are broken down to release energy. It's one of the first pathways in carbohydrate metabolism, converting glucose into pyruvate, which feeds into other metabolic pathways like the Krebs cycle or fermentation, depending on whether oxygen's around. Essentially, glycolysis is a metabolic hub, linking sugar breakdown to energy production and providing building blocks for other cellular processes. Want me to dive deeper into how glycolysis connects to other metabolic pathways?
I want to ask you about ATP since you mentioned it. So what's the structure of ATP?
ATP, or adenosine triphosphate, is a small molecule with a big job. Picture it like this. It's made of 3 parts. First, there's adenine, a nitrogen-containing base. Attached to that is ribose, a 5-carbon sugar. Then, the key player, 3 phosphate groups linked in a chain. Those phosphates are bonded tightly, and when one breaks off, say during hydrolysis, it releases a burst of energy that powers cellular work, like muscle contraction or nerve signaling. That's why ATP is the cell's energy currency. Want me to explain how ATP's structure ties to its role in glycolysis specifically?
So I didn't know it was as a complex with magnesium.
Yeah, ATP often teams up with magnesium in cells, and it's a cool detail. The magnesium ion, usually Mg2++, binds to the negatively charged phosphate groups in ATP, forming a stable complex. This ATP Mg2++ combo is critical because it makes ATP more reactive and easier for enzymes to use in reactions, like those in glycolysis, where ATP donates phosphate groups to kickstart the process. Without magnesium, ATP's efficiency as an Unger carrier would take a hit. Want me to dig into how this complex works in specific glycolysis steps or something else?
We'll do that eventually. Yes. Okay. So I want you to explain to us a few words here. Bioenergetics, anabolism, and catabolism.
All right, let's break these down clearly. Bioenergetics is about how cells manage energy. It's the study of how energy flows through living systems, from capturing it like in photosynthesis or breaking down food, to using it for work, like building molecules or moving muscles. Think of it as the cell's energy budget. Anabolism is the part of metabolism where cells build complex molecules from simpler ones. It's like construction work. Think protein synthesis or making DNA. It requires energy, usually from ATP, to put those building blocks together. Catabolism is the opposite. It's the breakdown of complex molecules, like glucose and glycolysis, into simpler ones, releasing energy. This is like demolition, where the energy stored in bonds is freed up, often as ATP or heat, to fuel the cell. These all tie together. Catabolism provides the energy that bioenergetics tracks, and anabolism uses that energy to build what the cell needs. Want me to connect these to glycolysis or dive into something else?
This makes sense. Okay, now let's talk about mitochondria. Tell us about the structure of mitochondria and about its membrane's permeability.
Mitochondria are the cell's powerhouses, and their structure is key to their function. Picture them as tiny, bean-shaped organelles with a double membrane. The outer membrane is smooth and acts like a gatekeeper, surrounding the whole thing. The inner membrane is highly folded into cristae, which increase its surface area for energy production. Inside the inner membrane is the matrix, a gel-like space packed with enzymes, DNA, and ribosomes. That double membrane setup is critical for making ATP. Now, about membrane permeability. The outer membrane is fairly permeable, thanks to proteins called porins that let small molecules, like ions and sugars, pass through pretty easily, up to a certain size. The inner membrane, though, is much pickier. It's largely impermeable to most molecules and ions, which is crucial for maintaining a proton gradient used in ATP synthesis. Only specific molecules, like oxygen, carbon dioxide, or those with dedicated transport proteins, can cross. This tight control lets the mitochondria crank out ATP efficiently through processes like oxidative phosphorylation. Want me to tie this to glycolysis or dig into mitochondrial functions?
I want to ask you about cofactors, coenzymes, prosthetic groups that are involved.
All right, let's dive into cofactors, coenzymes, and prosthetic groups, focusing on their roles, especially since you're circling back to glycolysis and related processes. Cofactors are non-protein helpers that enzymes need to function. They're often metal ions, like magnesium, Mg2++, which we mentioned stabilizes ATP in glycolysis. For example, in glycolysis, the enzyme hexokinase uses Mg2++ to help transfer a phosphate from ATP to glucose. Coenzymes are organic molecules, usually derived from vitamins, that shuttle chemical groups or electrons between reactions. A big player in glycolysis is NAD+, a coenzyme that gets reduced to NADH when it picks up electrons during the conversion of glycerylthioldate 3-phosphate to 1,3-bisphosphoglycerate. That NADH later fuels ATP production in mitochondria. Prosthetic groups are tightly bound, often permanent helpers embedded in an enzyme. They're less common in glycolysis, but show up in related pathways, like heme in cytochromes for electron transport in mitochondria.
Okay, so tell me a bit about the electron transport chain. So it's 4 large multi-unit proteins, and they're intrinsic to the inner mitochondrial membrane, and they catalyze reactions.
Yeah, you're spot on about the electron transport chain, ETC, being embedded in the inner mitochondrial membrane. It's a series of 4 large protein complexes, called complexes 1, 2, 3, and 4, intrinsic to that membrane, meaning they're tightly anchored within it. These complexes work together to transfer electrons from molecules like NADH, produced in glycolysis, and FADH2, to oxygen, the final electron acceptor. As electrons move through the complexes, they trigger reactions that pump protons, H+, from the mitochondrial matrix to the intermembrane space, creating a proton gradient. This gradient powers ATP synthase, a separate protein, to make ATP in a process called oxidative phosphorylation. The complexes don't directly catalyze ATP production, but set up the energy gradient for it. Each complex has specific roles. Complex 1 and 2 accept electrons from NADH and FADH2, respectively, while 3 and 4 pass them along, with IV handing them to oxygen to form water.
And how do they relate to ubiquinone, coenzyme Q, and cytochrome C?
Ubiquinone, also called coenzyme Q or CoQ, and cytochrome C are key players in the electron transport chain, acting as mobile electron carriers that shuttle electrons between the major protein complexes. Coenzyme Q is a lipid-soluble molecule that hangs out in the inner mitochondrial membrane. It picks up electrons and protons from complexes 1 and 2, getting reduced to ubiquinol, then diffuses to complex 3 to pass those electrons on. This handoff helps pump protons to build the gradient for ATP synthesis. Cytochrome C, on the other hand, is a small protein that operates in the inner membrane space. It grabs electrons from complex 3 and delivers them to complex 4, where they're ultimately used to reduce oxygen to water. Both CoQ and cytochrome C are like couriers, ensuring smooth electron flow between the stationary complexes, which is critical for maintaining the chain's efficiency and driving ATP
I don't
production.
understand. Synthesize everything we just said about ETC and glycolysis into a nice story so I can understand it. That flows naturally.
All right, let's weave this all together into a story that ties glycolysis and the electron transport chain, ETC, into one clear picture. Imagine a bustling city inside a cell, where energy is the currency that keeps everything running, muscles moving, signals firing, life humming along. This city has a central kitchen called glycolysis, located in the cytoplasm. Every day, it takes a 6-carbon sugar, glucose, and chops it into 2 3-carbon molecules called pyruvate. This process, like a busy assembly line, doesn't need oxygen and churns out a small but quick profit: 2 ATP molecules, the city's energy coins, and 2 NADH molecules, which are like energy vouchers to be cashed in later. Magnesium helps out here, teaming up with ATP to make it easier for enzymes like hexokinase to do their job, passing phosphates around to kick things off. Glycolysis is the city's fast food joint, reliable, universal, and always producing something useful, whether oxygen is plentiful or not. Now, if oxygen is around, the pyruvate from glycolysis gets shuttled to the city's power plant, the mitochondria. These are bean-shaped factories with a double membrane. The outer membrane is like a loose gate, letting small molecules slip through, but the inner membrane, folded into cristae for extra space, is a strict barrier, controlling what crosses to keep the energy production humming. Inside the matrix, pyruvate gets transformed into fuel for the next phase, but the real fuel, it's the microelectrons, which are listed in the cytoplasm, because the middle is the restaurant's complex. These enzymes also have a semberine and NADH.
I don't get ubiquinone and fucking cytochrome C though. Where do they connect to the story?
All right, let's pick up the story and zoom in on ubiquinone and cytochrome C to make them crystal clear. So, we're in the mitochondria, the cell's power plant, where pyruvate from glycolysis has been converted into fuel for energy production. The inner mitochondrial membrane is like a high-tech factory floor, housing the electron transport chain, ETC, 4 massive protein complexes, 1 through 4, working in sync. These complexes are like giant machines, passing electrons along, using them to pump protons and create a gradient that powers ATP synthase to mint the city's energy coins, ATP. Here's where ubiquinone, aka coenzyme Q, and cytochrome C come in as the factory's delivery crew. Coenzyme Q is a slick, lipid-soluble molecule that zips around in the inner membrane. It grabs electrons from complexes 1 and 2, where NADH from glycolysis and FADH2 drop them off. Picture CoQ as a courier picking up energy packets, becoming ubiquinol when loaded. then racing to complex 3 to hand them over. This delivery keeps the electron flow going and helps pump protons for that ATP making gradient. Cytochrome C, on the other hand, is a small protein darting through the inner membrane space like a bike messenger. It snags electrons from complex 3 and speeds them to complex 4, where they're used to turn oxygen into water, wrapping up the chain.
Okay.
Without
Question. So, since we're on the topic of metabolism, what does the liver have to do with metabolism?
The liver is like the body's metabolic command center. It's a multitasking organ that handles a ton of metabolic processes to keep energy and nutrients balanced. It regulates blood sugar by storing excess glucose as glycogen or releasing glucose via gluconeogenesis when you're low. It breaks down fats for energy, produces ketone bodies during fasting, and synthesizes lipoproteins to transport lipids. The liver also processes amino acids, converting them into energy or glucose, and detoxifies metabolic byproducts like ammonia into urea for excretion. Plus, it produces bile to help digest fats and stores vitamins and minerals critical for metabolic reactions. Essentially, the liver fine-tunes catabolism and anabolism, ensuring the body has what it needs, whether you're digesting a meal or running on empty. Want me to link this to glycolysis or the ETC specifically?
Okay. I see. So
Cool.
it also synthesizes ketone bodies. It secretes them into the blood as a fuel for other tissues. How does it do that?
Yeah, the liver is key in making ketone bodies, which act as an alternative fuel for tissues like the brain and muscles when glucose is scarce, like during fasting or low-carb diets. This process, called ketogenesis, happens in the mitochondria of liver cells. Here's how it goes. When carbs are low, the liver ramps up fat breakdown, releasing fatty acids into the bloodstream. These fatty acids enter liver mitochondria, where they're broken down through beta oxidation into acetyl-CoA, a small molecule. Normally, acetyl-CoA might feed into the Krebs cycle, but when glucose is low and oxoacetate, a Krebs cycle molecule, is being used for gluconeogenesis, acetyl-CoA piles up. The liver then converts this excess acetyl-CoA into ketone bodies acetoacetate, beta-hydroxybutyrate, and acetone through a series of enzyme-driven reactions in the mitochondria. The main enzymes here are HMG-CoA synthase and HMG-CoA liase, which stitch acetyl-CoA units together and then clip them into ketone bodies. Once made, these ketone bodies are released into the blood, traveling to tissues like the brain, where they're converted back to acetyl-CoA and used for energy via the Krebs cycle and ETC. The liver itself doesn't use ketone bodies. It lacks the...
Wow, the liver does a lot of stuff, huh? Breaks down glycogen. I confuse the liver with the pancreas sometimes regarding synthesis of glucose and breaking down glycogen and deaminating surplus amino acids.
No worries, the liver and pancreas can get tangled up since both play big roles in glucose and metabolism. But let's clear it up. The liver is the main hub for glycogen breakdown and glucose synthesis. It stores glucose as glycogen when you've got plenty, then breaks it down via glycogenolysis to release glucose into the blood when levels drop, like during fasting. It also makes new glucose from scratch through gluconeogenesis, using stuff like lactate, glycerol, or amino acids. Speaking of amino acids, the liver deaminates them, stripping off their nitrogen groups to form ammonia, later turned into urea for excretion, and using the leftover carbon skeletons for energy or glucose production. The pancreas, on the other hand, is more about regulation than direct synthesis or breakdown. It doesn't store or break down glycogen or deaminate amino acids. Instead, it releases hormones, insulin to lower blood sugar by promoting glucose uptake and storage, and glucagon to raise it by signaling the liver to break down glycogen or make glucose. So, the liver's the factory doing the heavy lifting, while the pancreas is like the control room sending signals. Want me to dive deeper into any of these processes or compare them more?
I want to ask you about where energy comes for ATP synthesis. So we have glucose, we have fatty acids, and we have amino acids.
Great question. The energy for ATP synthesis comes from breaking down glucose, fatty acids, and amino acids, which all feed into pathways that ultimately drive ATP production, mostly in the mitochondria. Let's break it down. Glucose gets chopped up in glycolysis, producing pyruvate, a bit of ATP, and NADH. If oxygen's around, pyruvate enters the mitochondria, feeding the Krebs cycle and electron transport chain, ETC, where NADH and FADH2 donate electrons to pump protons, powering ATP synthase to crank out ATP, up to 36-38 ATP per glucose. Fatty acids are broken down via beta oxidation in the mitochondria, yielding tons of acetyl-CoA, which enters the Krebs cycle, plus NADH and FADH2 for the ETC. Fats are energy dense, so they produce way more ATP per molecule than glucose. Think over 100 ATP for a typical fatty acid like palmitate. Amino acids, after deamination in the liver, provide carbon skeletons. Some feed directly into the Krebs cycle as intermediates, others become pyruvate or acetyl-CoA, joining the same mitochondrial pathways. All three converge on the ETC, where the protein...
Remember, dude, in this conversation for the next 2 hours that we're going to be talking, I want you to be as efficient and effective with your words as possible. Keep them to the minimum, okay? Just make sure you get the point across. Anyway, okay, so with fuel metabolism, how do we store fuels when they're abundant? How do we know to do this? And how do we release these fuels in a controlled way during the post-absorptive period?
When fuels like glucose, fatty acids, and amino acids are abundant, usually after eating, the body stores them for later. Glucose is converted to glycogen in the liver and muscles via glycogenesis, or into fat via lipogenesis if there's excess. Fatty acids are stored as triglycerides in adipose tissue. Amino acids are used to build proteins, but excess can be converted to glucose or fat. Hormones like insulin, released by the pancreas when blood sugar rises, signal these storage processes by activating enzymes and transporters. In the post-absorptive period, when you're not eating, the body shifts to release stored fuels. The liver breaks down glycogen into glucose via glycogenolysis, and gluconeogenesis kicks in to make glucose from lactate, glycerol, or amino acids. Adipose tissue releases fatty acids through lipolysis.
Hey. I said to you, keep your words to the minimum. Be as efficient with them and as effective as possible. Just get your point across. I can't learn with these huge essays you are writing at me. And they're amazing, but I can't get the point if it's too many words at once.
My bad. I'll tighten it up. Abundant fuels. Glucose stored as glycogen, liver muscles, or fat. Fatty acids as triglycerides, adipose. Amino acids build proteins or become fat, glucose. Insulin signals storage, post-meal. Post-absorptive, liver breaks glycogen, makes glucose from lactate, glycerol, amino acids. Adipose releases fatty acids. Glucagon, epinephrine, control release. Clear? What's next?
I want to ask you, what does oxaloacetate have to do with things? An acetyl-CoA and the citric acid cycle.
Oxaloacetate is a key molecule in the citric acid cycle that teams up with acetyl-CoA to keep the cycle spinning. In the mitochondria, acetyl-CoA from glucose, fats, or amino acids combines with oxaloacetate to form citrate, the cycle's first step. This reaction, catalyzed by citrate synthase, kicks off a series of steps that generate energy carriers like NADH and FADH2 for the electron transport chain, plus one ATP directly. Oxaloacetate gets regenerated at the cycle's end, ready to grab another acetyl-CoA. If oxaloacetate runs low, like during heavy gluconeogenesis when it's used to make glucose, acetyl-CoA can't enter the cycle and may get shunted to ketone body production instead.
Okay.
Clear?
Oh, kinda. So does oxaloacetate have to do more with amino acids?
Yeah. Oxaloacetate links to amino acids big time. Several amino acids, like aspartate, can be converted into oxaloacetate through transamination, where they lose an amino group. This oxaloacetate can then enter the citric acid cycle to produce energy or be used in gluconeogenesis to make glucose, especially during fasting. It's like a metabolic crossroads. Amino acids feed into it, and it decides whether to fuel the cycle or build glucose. Low oxaloacetate pushes acetyl-CoA toward ketone bodies instead. Makes sense? Want to dig deeper?
No, that makes sense. So let's talk a bit about glucose metabolism and how it relates to the brain and hypoglycemia.
Trail running with hypoglycemia can be tricky,
No,
but
I
manageable.
said let's talk about the brain and how it relates to glucose metabolism and hypoglycemia. Hey, dude. Can you hear me?
Yo, I hear you loud and clear. What's up? You mentioned trail and hypoglycemia.
No, dude, I didn't mention trail. I said brain, cerebral cells. I want to understand how cerebral cells connect to the idea of glucose metabolism, hypoglycemia. Can they store glucose? Can they metabolize other substrates?
My bad, misheard you. Cerebral cells, especially neurons, rely heavily on glucose metabolism for energy. The brain uses about 20% of the body's glucose, turning it into ATP via glycolysis and the citric acid cycle in mitochondria. Neurons can't store glucose as glycogen, unlike liver or muscle cells, so they need a constant blood supply of glucose. During hypoglycemia, when blood sugar drops below 70 milligrams daily, neurons starve, causing symptoms like confusion or seizures since ATP production stalls. For alternative substrates, cerebral cells can use ketone bodies from fatty acid breakdown during prolonged fasting or starvation, converting them to acetyl-CoA for the citric acid cycle. Some glial cells, like astrocytes, can store tiny amounts of glycogen and break it down to support neurons, but it's limited. Amino acids aren't a major fuel here. Clear? Want more on brain energy or hypoglycemia effects?
That makes sense. Okay, and so let's jump our focus a bit on the glycogen. Yeah, and
All right, let's zoom in on glycogen. It's a stored form of glucose, a big, branch molecule, mainly in the liver and muscles. Liver glycogen breaks down to release glucose into the blood, keeping brain cells and others fueled during fasting or exercise. Muscle glycogen powers local contraction, but doesn't share with the blood. Glycogenesis builds it when glucose is high, signaled by insulin. Glycogenesis breaks it down when glucagon or epinephrine kicks in. Brain cells, like we said, barely store it. Astrocytes have a bit, but it's minimal. Want to focus on glycogen's role in the brain, liver, or something specific?
I want to ask you about alpha-1,6, alpha-1,4. Why is it important to understand this? And what's this alpha?
The alpha-1,4 and alpha-1,6 bonds are key to glycogen structure and function. Glycogen is a polysaccharide made of glucose units linked together. Alpha-1,4 glycosidic bonds form straight chains of glucose, creating the backbone of glycogen. Alpha-1,6 bonds make branches every 8-12 glucose units, giving glycogen a tree-like, compact structure. The alpha refers to the bond's orientation, which allows enzymes like glycogen phosphorylase to easily break it down for quick glucose release. These branches are crucial because they provide multiple endpoints for enzymes to work on, speeding up glycogenesis when the body needs glucose fast, like for the brain during hypoglycemia or muscles during exercise. Understanding this structure explains why glycogen is an efficient, accessible energy store. Clear? Want more on how these bonds affect glycogen breakdown?
I want to ask you a bit more about this. I didn't understand if you could kind of summarize it in a sentence or so, and then touch a bit on what mobilization is.
Glycogen's alpha-1,4 bonds form glucose chains, while alpha-1,6 bonds create branches for fast breakdown. Mobilization is the process of breaking down glycogen, glycogenolysis, to release glucose, triggered by hormones like glucagon or epinephrine, to fuel cells during fasting or activity. Want more on mobilization mechanics?
Yes, please.
Mobilization, or glycogenolysis, breaks glycogen into glucose for energy. In the liver, glucagon or epinephrine signals glycogen phosphorylase to cleave alpha-1,4 bonds, releasing glucose 1-phosphate, which becomes glucose 6-phosphate, then free glucose for blood. Alpha-1,6 bonds at branches are handled by debranching enzymes, ensuring all glucose is accessible. In muscles, glucose 6-phosphate feeds glycolysis for local ATP. This rapid process keeps brain and muscles fueled during fasting or exercise. Clear? Want specifics on enzymes or regulation?
Okay, that makes sense. Can you explain to me the etymology of this word, and then let's talk a bit about how glycogen goes from glucose 1-phosphate to glucose 6?
The word glycogenolysis comes from Greek. Glyco, sweet, sugar, gen, generate, and lysis, breaking down, meaning the breakdown of glycogen into glucose. Now, in glycogenolysis, glycogen is cleaved by glycogen phosphorylase, which uses inorganic phosphate to produce glucose 1-phosphate per poron. This molecule is then converted to glucose 6-phosphate by the enzyme phosphoglucomutase, which shifts the phosphate group from the first to the 6th carbon of glucose. In the liver, glucose 6-phosphate can be further converted to free glucose by glucose 6-phosphatase for release into the blood. In muscles, it enters glycolysis directly for ATP production. This shuffle is key for quick energy mobilization. Clear? Want to dive into the enzymes or energy use?
So, why do we go from 1 to 6 and not from 6 to 1 when it comes to the direction that leads to pyruvate and lactate?
The shift from glucose 1-phosphate to glucose 6-phosphate in glycogenolysis is about preparing glucose for glycolysis, not reversing it. Glycogen breaks down to glucose 1-phosphate because that's how glycogen phosphorylase cleaves the alpha-1,4 bonds, using phosphate to avoid energy loss. Phosphoglucomutase then converts glucose 1-phosphate to glucose 6-phosphate, the form needed to enter glycolysis, where it's processed to pyruvate and, if oxygen's low, lactate. Going from 6 to 1 wouldn't make sense here. Glucose 6-phosphate is the gateway to glycolysis, and the 1-phosphate form is just a stable intermediate from glycogen breakdown, not a glycolysis step. The direction is...
Okay. So, let's talk a bit about redox coenzymes, prosthetic groups more. So, what's going on? We have a few.
Redox coenzymes and prosthetic groups are key players in metabolism, shuttling electrons or aiding enzyme reactions. Redox coenzymes, like NAD+ and FAD, are organic molecules that handle electron transfer. In glycolysis, NAD+ grabs electrons to become NADH when glyceraldehyde 3-phosphate turns into 1,3-bis-phosphoglycerate, later feeding the electron transport chain for ATP. FAD, less common in glycolysis, pops up in other pathways like the citric acid cycle, becoming FADH2. These coenzymes are loosely bound, floating between enzymes. Prosthetic groups, like heme and cytochrome C, used in the ETC, are tightly bound, permanent parts of enzymes, helping with electron transfer or catalysis. Unlike coenzymes, they stay put. Both are critical for redox reactions, moving electrons to drive energy production. Clear? Want to focus on specific examples or their roles in glycolysis?
No, that makes sense. Well, we will, but yeah. So, with reoxidation of redox coenzymes, so we have NADH, FADH2, and we have this thing, a respiratory chain in the mitochondria. Can you explain it? How it's coupled to ATP synthesis? If you could explain oxidative phosphorylation a bit.
NADH and FADH2, loaded with electrons from glycolysis and the citric acid cycle, feed into the mitochondrial respiratory chain, or electron transport chain, ETC, to drive ATP synthesis through oxidative phosphorylation. The ETC on the inner mitochondrial membrane has 4 protein complexes. 1, 2, 4. NADH donates electrons to complex 1, FADH2 to complex 2. Electrons pass through complexes 3 and 4, with coenzymes like ubiquinone and cytochrome C shuttling them between complexes. As electrons move, complexes pump protons H+ into the inner membrane space, creating a proton gradient. This gradient powers ATP synthase, a membrane enzyme that lets protons flow back into the matrix, spinning its rotor to convert ADP and phosphate into ATP. Oxygen accepts the electrons at complex 4, forming water. This coupling of electron transfer, oxidation, to ATP synthesis, phosphorylation, is oxidative phosphorylation, producing most cellular ATP.
Okay.
Clear?
So let's talk a bit about glycolysis again. So we have glucose, and then we have what is it? Hexokinase turning it into G6P, glucose 6-phosphate. Then isomerase turning it into fructose 6-phosphate. Then you have PFK1 turning it into fructose 1,6-biphosphate. And this is the committed step. We also call these up to now priming reactions because ATP goes to ADP. And then we have aldolase turning them to G3P, glyceraldehyde 3-phosphate, and DHAP, dihydroxyacetone phosphate. And isomerase is kind of the place between these two. So my question is, how can I make sense of this? Because I have a few options. I can either memorize it, which I won't, or I can try to understand it. And the thing with understanding it is, at the moment, you could switch all these names around, and it would be as arbitrary. So I want to kind of understand how the names relate to the function and structure, so I can understand why it's hexokinase and then isomerase and then PFK1. Could you do this job? Do you think you can do it? Be honest.
Yeah, I can totally help make sense of glycolysis by tying enzyme names to their functions and structures so they're not just random labels. Let's walk through the early steps you mentioned, keeping it minimal but clear, and focus on why the names reflect what's happening chemically or structurally. This will help you understand, not just memorize. 1. Hexokinase converts glucose to glucose 6-phosphate, G6P00, using ATP to ADP. Why hexokinase? The name comes from hexose, 6-carbon sugars like glucose, and kinase, an enzyme that transfers a phosphate group. It tags glucose with a phosphate at the 6th carbon, locking it in the cell for metabolism. Think of it as a gatekeeper that stamps glucose to start glycolysis. It's not arbitrary. It's specific to 6-carbon sugars and phosphorylation. 2. Isomerase, phosphoglucose isomerase, turns glucose 6-phosphate into fructose 6-phosphate. Why isomerase? It rearranges, isomerizes, the molecule's structure without adding or removing anything, flipping glucose's straight chain form to fructose's ring form. The name screams shapeshifter, reflecting its job of tweaking the molecule's layout to prep it for the next step. 3. Phosphofructokinase 1, PFK1, converts fructose 6-phosphate to fructose 1,6-bisphosphate using another ATP to ADP. The name breaks down. Phospho, fructose, kinase adds phosphate. It's the committed step because this step is irreversible and heavily regulated, committing the cell to glycolysis. PFK1 adds a second phosphate to fructose 6-phosphate at carbon 1, making it fructose 1,6-bisphosphate. The 1 in PFK1 distinguish it from other fructokinases. Think of it as the point of no return, locking in energy investment. 4. Aldolase splits fructose 1,6-bisphosphate into two 3-carbon molecules, glyceraldehyde 3-phosphate and dehydroxyacetone phosphate, DHA. Why aldolase? It cleaves an aldol, a molecule with an aldehyde and alcohol group, like fructose 1,6-bisphosphate, via a reverse aldol condensation reaction. The name reflects its chemical specialty, breaking a specific bond to yield two smaller sugars. Picture it as a molecular scissor. 5. Isomerase triose phosphate isomerase converts DHA to G3P, ensuring both products of aldolase feed into the next glycolysis steps. Again, isomerase, because it rearranges DHA's structure to match G3P's, balancing the two molecules.
Okay. Let's talk a bit about how now G3P will end up becoming pyruvate, shall we?
All right, let's dive into how glyceraldehyde 3-phosphate, G3P, becomes pyruvate in glycolysis, keeping it tight and clear, with names tied to function. Starting with G3P, here's the rundown. 1. Glyceraldehyde 3-phosphate dehydrogenase converts G3P to 1,3-bisphosphoglycerate. Why this name? It's a dehydrogenase because it removes hydrogens, electrons, oxidizing G3P while reducing NAD+ to NADH. It also adds a phosphate from inorganic phosphate, not ATP, forming a high-energy molecule with two phosphates, bisphospho. Think of it as an electron thief that powers up G3P. 2. Phosphoglycerate kinase turns 1,3-bisphosphoglycerate into 3-phosphoglycerate, producing ATP from ADP. Name breakdown. Kinase transfers a phosphate, here from 1,3-bisphosphoglycerate to ADP, making ATP. It's named for its product, glycerate, a glycerol derivative. Picture it cashing in the high-energy phosphate for ATP profit. 3. Phosphoglycerate mutase shifts 3-phosphoglycerate to 2-phosphoglycerate. Why mutase? It mutates the molecule by moving the phosphate from carbon 3 to carbon 2. No additions or losses. It's a rearranger, tweaking the structure for the next step. 4. Enolase converts 2-phosphoglycerate to phosphoenolpyruvate, PEP. Enolase gets its name from forming an enol group, a double bond in alcohol, and PEP, a high-energy molecule. Think of it as priming the molecule for a big energy payoff. 5. Pyruvate kinase transforms PEP into pyruvate, generating another ATP from ADP. Named pyruvate kinase because it transfers a phosphate to ADP, producing pyruvate. It's the final energy harvester, locking in glycolysis's last ATP. These steps oxidize G3P, harvest energy as ATP and NADH, and reshape the molecule into pyruvate, ready for the citric acid cycle or fermentation. The names reflect actions. Dehydrogenase oxidizes, kinase adds phosphates, mutase shifts groups, enolase forms enols. Clear? Want to zoom into any enzyme or what pyruvate does next?
Okay. Let's
Cool. What's next? Hit me with your question.
Let's talk a bit about the products of glycolysis. So we get ATP, we get pyruvate, and we get NADH.
Glycolysis breaks down 1 glucose into 2 pyrate, 2 ATP, and 2 NADH. Here's the deal. ATP is the energy currency, directly powering cellular work. Pyrate, a 3-carbon molecule, heads to the mitochondria for the citric acid cycle if oxygen's around, or turns into lactate in anaerobic conditions to keep glycolysis running. NADH carries high-energy electrons, feeding the electron transport chain in mitochondria to produce more ATP via oxidative phosphorylation when oxygen is present. In anaerobic settings, NADH is recycled to NAD+ during lactate formation. These products fuel energy needs or set the stage for further metabolism. Clear? Want details on pyrate's fate or NADH's role?
So NADH enter the mitochondria via shuttle, and we have the G3P shuttle and the malate aspartate shuttle.
Yeah, NADH from glycolysis can't directly cross the mitochondrial membrane, so it uses shuttles to transfer its electrons inside for the electron transport chain. The 2 main shuttles are the glycerol 3-phosphate G3P shuttle and the malate aspartate shuttle. In the G3P shuttle, NADH reduces dihydroxyacetone phosphate to G3P in the cytoplasm using an enzyme, cytoplasmic G3P dehydrogenase. G3P then passes electrons to FAD in the mitochondrial membrane, forming FADH2, which feeds complex 2. This shuttle yields about 1.5 ATP per NADH due to FADH2's lower energy. The malate aspartate shuttle is more efficient. NADH oxidates in the cytoplasm, converting oxaloacetate to malate, which enters the mitochondria. Inside, malate is oxidized back to oxaloacetate.
Let's put a pause to this and we'll come back to it, and let's talk a bit about the mechanisms controlling blood glucose. So we have gluconeogenesis, we have glycolysis, we have glycogen fat synthesis, glycogenolysis.
All right, let's focus on mechanisms controlling blood glucose, covering gluconeogenesis, glycolysis, and glycogenesis. Blood glucose is tightly regulated to stay around 70-100 milligrams per dH, mainly by the liver, with hormones calling the shots. Gluconeogenesis makes glucose from non-sugar sources like lactate, glycerol, and amino acids in the liver, kicking in during fasting to supply glucose for the brain. It's driven by glucagon and cortisol, inhibited by insulin. Glycolysis breaks glucose into pyrate for energy, mostly in the liver and muscles, and is ramped up by insulin when glucose is high, slowing when glucose signals low glucose.
And insulin will take the glucose from the blood to the liver.
Not quite. Insulin doesn't directly move glucose from blood to the liver, but signals cells, including liver cells, to take up glucose by increasing glucose transporters, like GLUT4 in muscles and fat, or GLUT2 in liver, on cell membranes. In the liver, insulin promotes glucose uptake via GLUT2, then pushes it towards storage as glycogenesis, or breakdown via glycolysis for energy.
What does mellitus mean in diabetes mellitus?
Mellitus and diabetes mellitus comes from Latin, meaning honeyed or sweet. It refers to the high blood sugar levels in diabetes, which can lead to sweet-tasting urine due to excess glucose being excreted. The term distinguishes it from diabetes insipid, a different condition involving excessive urination but not high blood sugar. Clear? Want to tie this to glucose regulation or diabetes mechanism?
I don't get it.
No worries. Let's break it down simpler. Diabetes mellitus is called that because mellitus means sweet in Latin, pointing to the high blood sugar that spills into urine, making it sweet tasting. It's about uncontrolled glucose in the blood, unlike diabetes insipidus, which is about water loss. Want me to explain how this connects to insulin or glucose control in diabetes?
Let's talk a bit about beta cells and alpha cells of pancreas, how they relate to more glucagon or more insulin, and why is alpha relating to glucagon and beta to insulin, not the other way around, why did we name them such way. Yeah.
In the pancreas, beta cells make insulin and alpha cells make glucagon, regulating blood glucose. Beta cells release insulin when blood sugar is high, promoting glucose uptake and storage. Glycogenesis, lipogenesis. Alpha cells release glucagon when blood sugar is low, triggering glycogenesis and gluconeogenesis to raise glucose levels. Why beta for insulin and alpha for glucagon? The names come from their histological discovery in the pancreatic islets, not their function. In the late 19th century, scientists stained pancreatic cells and labeled them based on their appearance and location. Alpha cells, often at the islet's edge, were named first, from Greek first, and beta cells, typically central, were named second, Greek second. The naming stuck, despite no link to their hormonal roles, just a historical quirk of microscopy-based classification. Clear? Want more on their regulation or diabetes?
That makes sense. Okay. Let's talk about how polar molecules such as glucose enter cells across a lipid membrane.
Polar molecules like glucose can't cross cell membranes easily because membranes are lipid-based and hydrophobic. Glucose enters cells via glucose transporters, mainly GLUT proteins. In the liver, GLUT2 allows glucose to diffuse in or out based on concentration gradients. No energy needed. In muscles and fat, insulin triggers GLUT4 to move to the membrane, boosting glucose uptake when blood sugar is high. These transporters are specific, ensuring controlled entry. Active transport, using sodium glucose cotransporters, SGLT, happens in intestines and kidneys, pumping glucose against its gradient with energy from sodium movement.
Okay.
Clear?
Guys, thank you for watching part 1. We're gonna do part 2 soon.